So I'm Rebecca Thomas, I'm Research Project Manager at Sheffield Children's Hospital. Well, at the moment in the UK, newborn screening only uses biochemical analyses as a screening test, and so it really just limits the newborn screening programme to disorders with biochemical markers. And whilst this works really well for the current disorders, it does limit the newborn screening programme if it ever wanted to expand to include other disorders. So developing and optimising a genetic screening test just gives newborn screening increased flexibility, gives it an extra tool for their toolkit, so that they can consider disorders that may be added to the newborn screening programme in future, which don't have a biochemical marker but have a genetic marker. So we could run 2 96-well plates simultaneously, and over the course of like a 7-day working week, this means that you could run 1,000 samples per week. And screening labs in the UK really vary in terms of the numbers of samples that they receive. But for example, Sheffield Newborn Screening Lab receive about 1,000 samples a week. So proving that we had a protocol that could analyse 1,000 samples a week was really helpful. Yeah, so 1,000 samples a week is based on having one of everything. So one biomech robot platform and one Iron Chef and one S5 sequencer. So if you double those machines, and that means you could double your capacity. So you'd be doing 2,000 samples per week, for example. So we're analysing dry blood spots. So dry blood spots at the moment are they're blood taken on a Guthrie card, so they're collected from the heel of a baby on day 5, and at the moment that's because that's when biochemicals in the blood stabilise. With genetic testing, we can potentially take that dry blood spot on day 0, the day the baby's born. We get the dry blood spots and we punch it in an automated way using a machine which is just like a really fancy hole punch. It's got a barcode on it, so it screens a barcode associated with the dry blood spot into a 96-well plate. We can then take the 96-well plate and put it on a robot which will do the DNA extraction for us. And then we've also automated the rest of the protocol, which involves AmpliSeq library preparation. We do QC using the tape station, and then it's just a case of loading it on the Iron Chef and the day after the sequencer. At the most a day and a half, and that's mostly just waiting for the machine to do its thing, so it's very little hands-on time. I'd say sort of 15 minutes in terms of so it's less than 60 minutes for the dry blood spot punching. It's about 15 minutes hands-on time in terms of the library preparation for each step, and then also about 15 minutes for the Iron Chef and 15 minutes for the sequencer. The turnaround time of the workflow is 4 days. So on the first day you can achieve the punching of the dry blood spot, the DNA extraction and starting the library prep. On day 2 you finish the library prep, and then day 3 is the sequencing, and day 4 is the analysis and reporting. So the gene panel was a Iron AmpliSeq custom panel. So we used the AmpliSeq website to design this. So you can add the genes that you want onto the panel, and other little bits like the exon padding, for example.